biologists are obsessed with diversity. It's probably the primary characteristic of the biological mind, unlike the physical mind, that tends to be interested in unity. And so if you look across fields in biology, diversity, by which I mean here simply counting numbers of types operationally. Geneticists are interested in the diversity of regulatory networks, gene networks. Developmental biologists, the development of morphological varieties, body forms. Ecologists, like this guy, fascinated in the structure of trophic webs and communities and networks, through to evolutionary biologists who are somewhat obsessed with trees, inferring phylogenies. And the question that you naturally ask, I think, in biology is there some larger picture that would unify all of these approaches to diversity? And what's the connection between genetics and development, say, or evolution and ecology? And there are fields that sit at the intersection. Right? Evo-Devo, right, in some senses this one, and the field of evolutionary ecology and so on. Now, I couldn't resist putting this up. This is Enrico Fermi, a physicist, who wrote this wonderfully rude remark about biologists. If I could remember the name of all these particles, I would be a botanist. And that divide still exists. I mean, many of my colleagues are theoretical physicists, and they're horrified by the biologist's interest in remembering and enumerating large numbers of species. There have been attempts to integrate across those different fields, and I just mentioned this because one of my colleagues at the Santa Fe Institute, Douglas Irwin, who's been working with Eric Davidson at Caltech, trying to look at how, and I won't talk too much about this, how the underlying gene regulatory networks change across deep time. So from the Cambrian, about 550 million years ago, to the present. So there are attempts to relate causally gene networks to morphology. But that's not what I mean by an integrated picture. I'll try to explain to you what I mean by that. So that is there a fundamental selective principle of the kind that Charles Darwin talked about? One spoke about at length yesterday, and I'll show you some more pictures of Charles in my talk. Not a mechanical process that leads to morphological variability, but is there a sort of framework, a mathematical framework, that can encompass all the kinds of diversity that I showed you in that first picture? And so from weird animals like the platypus, through to community structure like these bacterial mats, through to ecosystems. The wonderful property of the evolutionary theory, it's kind of material independent. It doesn't work at any one level, it works at all levels. And I'm going to claim, and you might not believe me, that the clue to this unified approach to diversity at multiple scales comes from the analysis of things like this, living forms of architecture. So let's just start with Darwin. So here he is. This is a beautiful photograph of him. I didn't believe it was real when I first saw it. Apparently it is. Here's The Origin of Species, written in 1859. So you asked the question, what is evolution? I'll tell you operationally. Evolutionary theory tries to explain correlations between organisms and their environment. Okay? And what makes evolution special is it invokes three processes at least. Right? Selection, differential survival, which he introduced, heredity, some form of transmission of the information, and replication. And so any time you invoke those three processes to explain correlations between characteristics of an organism and an environment, you're basically providing an evolutionary explanation. That's the definition of an evolutionary explanation. So it's not just change or transformation, it's a red herring. Okay. And so that's what he introduced in this book in 59. So the key to Darwinian thinking is to explain variability in organisms in terms of variability in the environment. Okay. And this is the famous tangled bank that Darwin spoke about in The Origin of Species. But Darwin wrote two other books that are less read. One of his first books was on the formation of coral reefs. And the last book that was published the year he died, the formation of vegetable molds by the action of earthworms. And these books invert the causal sequence that he introduced in The Origin of Species. Instead of explaining the form of organisms in terms of the form of the environment, he now explains the origin of the environment in terms of the action of the organism. It's a very different approach, because in some sense, if the organism builds the environment, the organism builds the selection pressure. So there's a peculiar circularity in the evolutionary dynamic, and that's what I'm going to be spending most of my time talking about. This weird world where your form is determined by your behavior via the environment that you build. Since I'm here in Valparaiso, I thought I'd mention this quote that came from his voyages that he wrote in 1830. This is where he spent time in 1834. He wrote it quite a lot later. Where he says, "The beagle anchored late at night in the bay of Valparaiso. When morning came, everything seemed delightful. The climate felt quite delicious, which is the heavens so clear and blue." But before you interrupt, oh, I think he can't see you because of the camera. Can you move? This guy can't see. Can you move that way, maybe back a little? There's a guy.
Un poquito más.
Is that better? Good. Yeah, but I thought, so you wouldn't feel too gratified, proud of that quote, I'd add this one. Where it said, "Valparaiso is not very productive to the naturalist." It was too much of a city for him, so he had to leave to find some beetles or other specimens that he found more interesting than humans. Now, this approach, where you study how organisms change their environment, rather than the environmental effect on the organism, really lay dormant for over a century. And then Carl von Frisch in 1974 wrote this very famous book, Animal Architecture. He'd won the Nobel Prize in 1973. It's the only Nobel Prize that ever went to behavior and evolution. He shared it with two other very prominent biologists, Lorenz and Tinbergen. But this book was really Frisch's attempt to demonstrate that human productions were not unique. That in fact animals were every bit as impressive as humans in constructing glorious edifices. So it was much more in the service of a worldview that wanted to diminish the differences between humans and the rest of animal nature. These books start getting more interesting from the conceptual point of view. These were two published in 1982, one by Richard Dawkins. This is The Extended Phenotype. It is his best book. The Selfish Gene is perhaps the best known. The Extended Phenotype is the most admired, at least by academics. And Dick Lewontin's The Triple Helix. In these books, what they ask is the following. They say, the action of the gene doesn't terminate at the boundaries of the body. It extends into the environment. So the concept of adaptation has to be modified to accommodate that observation. And so they were explorations of the implications of that insight. It's a horrible phone. This is a horrible phone. And so then finally, mine is a nice Japanese flute. And then finally, this book, which is perhaps the most extensive development of the concept in mathematical terms, was written by John Oglinsky, Kevin Lane, and one of my colleagues at the Institute, Mark Feldman, who's also at Stanford, where they call this process, whereby organisms modify the environment, niche construction. Okay? So I need to tell you a little bit about niche construction. First I need to tell you about the niche. Now the niche is one of those, biologists are very fond of using terms that are very hard to define. And there are good and bad reasons for that. But the niche is one of those things where in an elementary biology class, you'll spend a lot of time arguing about what it means. It started with Grinnell in 1917, and he was basically saying organisms depend on some property of the environment that they consume free energy from. And I'm going to call that the niche. It was very vague. What Elton added to this was that the environment wasn't just abiotic, it was constituted of other organisms. So whatever your niche definition was, you'd have to also factor in other individuals who were feeding them. Hutchinson tried to make it a little bit more technical by calling it a high-dimensional space of combined organism-environment interactions. Okay. So it's not that different from Milton, it's just he introduces the concept of dimensionality, but he did it qualitatively. Now, niche construction is really how that complement of organism and environment comes into existence. Okay. Most mathematical models in ecology assume the existence of an environment and assume the existence of species, and then they monitor the consequences through time of the interaction. Niche construction tries to explain how they came into existence. So it has an ecological dimension, it has an evolutionary dimension too, because you have to worry about adaptive radiation. The key concept here, formally, is that you can't separate the time scales of ecology and evolution, which is something that Hutchinson spent a lot of time discussing. He called it the evolutionary play and the ecological theatre. Because the play, of course, is played over many generations, but the place where it is performed and the actors assume the roles, assume it transiently. And niche construction questions this notion of the separation of timescales, because now the actors build the play. So this is a picture from the last book I mentioned, the niche construction book, where John Ogling Smee, it's actually from a paper he wrote in the book, where he makes a contrast between regular Darwinian evolution and niche construction. So the regular Darwinian evolution says the following: there's some environment that persists in time. This is generational time, running down. Natural selection operates on the gene pool, differentially biasing the frequencies of different genotypes. That's another, by the way, that was Ernst Mayr's definition of evolution, the change in time of gene frequencies. Much more operational. So natural selection is, in some sense, independent of what the organisms are doing. That's the key. Under niche construction, that's no longer true, because you'll see now that the gene pool influences the environment, hence it influences selection in its own future. Well, I live in now, I change the selection pressure for my descendants. So it's quite a different dynamic to the standard Darwinian dynamic. And clearly that's very much the dynamic of our own species. So just to make this a bit more concrete, here's a very simple pair of differential equations which an ecologist might write down to study the dynamics of competition. So you might have two species, let's call them x and y. Leibniz notations to dx by t, dt, and dy by dt. And through time, one species will, for example, competitively exclude another species. So this equation has the property of exclusion, Gauss's principle, such that one species persists in equilibrium and one is outcompeted. And the key here is that they differ in their replication rate and, for example, in their carrying capacity. That is the asymptotic density that they can survive at. But notice that the carrying capacity is not anything that these species can control. It's exogenous. At the end, I'll give you some references to papers if you choose to read them again. In niche construction, things change a little bit, and it's a simple schematic. You have two species again. They can replicate as before, but now they can make a decision. They can decide, maybe I won't replicate all the time. Maybe I'll invest in the environment. So I'll build. Okay? And that means you have to have a slightly more complicated set of equations, basically the same principle, but now you'll see that you can choose between, on the one hand, growing or reproducing, or investing. So with, let's say C varies between 0 and 1, I can do or replication, in which case we're back in the first equation, or I can actually invest in a niche. So there's a trade-off in these kinds of models between niche construction and regular replication. Because otherwise it would be cheating. You're not allowed to build an environment and not pay for it. And if you write down these mathematical models, I'm glossing over a lot of detail, but again, I have to download the paper if you like. Some interesting implications arise from building these slightly more complicated models. The first one is the assumption, right? But you also get this possibility of greater abundance, because I'm building an environment that didn't exist before, so I can more effectively exploit sources of free energy. You can show that you have, because of that, greater stability. If you look at the standard models of Bob May, I don't know how many are familiar with that, but if you look at spectral properties of the so-called community matrix, you can demonstrate that you get greater stability in such niche construction systems. And actually increased diversity equilibrium, which here simply means more species in the stable equilibrium. Okay. So these are some of the implications of niche construction. But there's a significant problem with the theory of niche construction. And it's interesting that it wasn't noticed by Dawkins or Lewontin or Mark and John and Kevin. And a little bit of maths. Here's the model again. It's one I showed you. You can write down many different models for niche construction, but you always have to assume some trade-off between growing and building. If you solve that system of equations, and you look at the relative rates of growth of two species or two strains that differ only in their tendency to niche construct or replicate, so you have C and C prime, that's the only way in which they differ, you'll see from the solution to this equation that the species that has a higher value of C, or C prime, hence a lower value of 1 minus C, right, will actually outcompete the other. So look, so if in this case, if C prime is greater than C, this is positive, so you have exponential divergence, right? That means that there's never any reason to niche construct. In biology, this is called the tragedy of the commons. We've all heard about this in terms of the evolution of cooperation and altruism. You know, if Pablo's going to build the niche, I'm not building it, I'll just replicate. Let him do it, because I can use it. And so there's a fundamental problem in niche construction, that there's no reason to niche construct if someone else will do it for you. And yet, you look all over the world, and there is animal architecture, there is niche construction. How is it possible when we know that fundamentally it's evolutionarily unstable? So it's a little bit like the problem of cooperation. And the key in niche construction is that you can monopolize the products of your construction. If your house that you build, for some reason you have preferential access to, and you can ensure that some portion of its benefits accrue only to you, then you're in business. So you have to somehow discover mechanisms for partitioning the niche into a large fraction which only you can exploit. Otherwise, nothing. And as I'm going to show you in a minute, otherwise really nothing. No bodies, no ecosystems. Because what I'm going to show you is that niche construction is the fundamental principle that also explains development, but we'll get there.
So, yeah. There's a question here. So in fact what you're doing is you are coupling, you are no longer treating reproduction and niche construction as trade-off. You are coupling both. So you when you reproduct, you niche construct somehow. And then that way you, eventually create this monopolization of what you do is for your use only.
Yeah, well, there's still a trade-off, but take a very simple example. Imagine it was spatial.
Okay.
So that if I, like a caddisfly larvae case, if I niche construct the case, I use energy to build it, but it's around my own body, you can't really use it very well. So in some sense, there are many ways in which I can restrict access to what I build. And one of the most obvious ones is spatial. With a bird nest, it's harder, for example, because another bird might come along and displace you. But there's still a trade-off. It's just you have to build this additional means of ensuring that only you benefit.
But you would say that a bird nest is niche, right?
Absolutely. I would. Absolutely. Because that object contributes to free energy sources. In fact, I thought almost anything that your genome contributes to constructing a part of your niche. So, let me just give you a little slide, a numerical slide, just to make this point again. So what this slide shows is the following. This is time. This is different values of monopolization. 100% monopolization. You get everything from the environment you build. You get about half of it, or you get nothing. Now in this case what happens, right, is because there's no environment built, no species can survive. I don't assume that there are environments in the world for you to occupy. Everything gets built by something, in which case you get nothing in equilibrium. Okay? No one niche constructs, the value of C is very high, so you only replicate. Right? And there's no niche. As you start increasing the value of M, your ability to monopolize your niche, then systematically you get increases in the mean value of niche construction, right? And more species can exist at equilibrium. So this is kind of obvious, right? I mean, if there's no environment, and then here again, this is with perfect monopolization. And notice, by the way, in this particular set of equations, that you still get competitive exclusion. You get a whole sequence of species which have different levels these happen to be different levels of C. And eventually it moves to the equilibrium, which is value of niche construction, which is given by 1 over 1 plus m. So you invest half of your energy in replication and half of your energy in niche construction. Okay? So this is pretty obvious. Now, if you look at an ecosystem, it doesn't look very much like an organism. Right? It's very distributed, you have a lot of variability, things seem very independent. When I look at an organism, look at my body, for example, it looks as if all my cells are working together in a coordinated fashion to produce a concerted pattern of behavior. Okay? What I want to tell you, or try and persuade you of, is that both of them exist on a continuum of niche construction. And what a body really is it's a perfect ecological monopoly. Okay? In fact, it's more like a monarchy as opposed to an ecological democracy. But fundamentally, dynamically, they're very similar. So how is that possible? So let's just look at a few cases. Here's if you're interested in ants or social insects, an ant colony. Ants have a variety of different castes. They live in these extraordinary structures that some of the species build. And here's a body. The body also has a variety of castes. We call them organs. We have many different types of organ system and function and many more cells. The fundamental difference in a body and a mini ecosystem like this social system is that this one is much more complicated or much more diverse. And Bill Hamilton in the 60s, when he got interested in the theory of kin selection, which is associated with him, felt that the most complicated forms in nature would be bodies, and the reason for that is because they would allow for the greatest differentiation without competition. So an ecosystem has limited complexity by that way of reasoning, because everything is trying to consume the resources of everything else, whereas in a body, the cells of your body have a kind of the germline in mind, and so they'll limit their competition. That's what he felt. I don't agree with that. But nevertheless, fundamentally, there's not a big difference between these two structures. There's a germline in this one, and there's a germline in this one, probably not indicated. And there are sterile casts, in the same way that there are sterile casts here, namely everything you see on the right. Because nothing here contributes to evolution in the long term. Your kidney doesn't replicate and generate a subsequent generation. Only your germ cells. Okay? So the body is an ecosystem, right? But it's an ecosystem more like the ecosystem of a social insect with many sterile casts. Now, in 1970, okay, let me take one other point, because I think this will be a little clearer. You'll notice, by the way, that the people who study this tend to be ethologists and evolutionary biologists and ecologists, where the people who study this tend to be developmental biologists and physicians. Why is that? There's this perception that there's a fundamental difference between development and evolutionary dynamics. Now, in the 1970s, this guy, Jean-Pierre Changeux, started getting interested in Darwinian processes in the formation of the brain. So instead of thinking about the brain as being constituted by a program in the genome, somehow there's selection in the brain, and that the environment you live in will influence the structure of your nervous system. And we know this to be true now, especially in the formation of the visual system. And he wrote this book later, quite a few years later, called Neuronal Man, where he explores fully the idea that neural development is Darwinian. In the 80s, Leo Bass wrote this book, which for some people becomes a Bible. It's a very interesting book. Once you read it, you sort of become a devotee of it. And it's not clear why, but I've seen it many times. I think it's because it's very hermetic. It's quite cryptically written, so you feel as if you're somehow one of the members of a special congregation when you finally understand it. But he also suggested that perhaps development was selective. Okay? That the cells in the body are in competition with each other, and that the final, if you like, equilibrium distribution that constitutes your morphology is the outcome of a local selection pressure operating during your lifetime between cells. And finally, and he's at Yale, and then Mark and John at Berkeley and at Harvard sort of amplified this in their notion of facilitated variation. If you want to talk about this, we can talk about it later. But again, this idea that development is selective. And one of the key reasons why they wrote this book is because if you talk to people who believe in intelligent design, which is a movement in the United States, and probably you know about it here too, where people claim that the perfection, the exquisite perfection of design suggests the requirement that some omniscient, omnipotent creator had a hand in it. And that if you were to change anything ever so slightly, the whole edifice would collapse. But if the developmental process is not programmed but selective, that's no longer true. Because if you perturb the system, then because it's competitive, something else that can do the job, maybe not as well, but pretty well, will take over. So development is stable precisely because it's Darwinian. Okay? Whereas if it was a program, like a computer program, and you put in an error, you get a syntax error, and it doesn't compile. So for them, in part, it was a means of refuting an argument that everything is perfect and perturbations are going to be lethal. So here's organismal development, here's how you normally see it represented schematically, cells dividing. But I'm just going to draw it in terms of something like a fate map. So now you have a germ cell that can divide into somatic cells, 1 minus C, or more stem cells with C. So this looks, by the way, a lot like the model I showed you before, where you can invest either in replication or the niche. So development is niche construction. Your germ cells are building a nest, which is your body. Richard Dawkins called this the vehicle, as opposed to the germline, which he called the replicator. So formally, they're almost identical. There's a bit of a difference, because you'll notice that these cells can also divide, whereas in the equations I showed you that wasn't true, but it doesn't matter. So development is a niche construction dynamic, with one kind of cell building an environment, but the key here is that the environment is almost perfectly monopolized. Because it's very difficult for me to use your soma. I hire people to use their soma. Parasites try and use your soma to replicate their germline. So there are attempts at hijacking the somatic tissue of other organisms, holding our own soma than an ant, for example, is at controlling its hive. And that's because of spatial distances and so forth. Right. I think it's important you understand this. Development is a special kind of ecosystem, a special kind of ecological dynamic. All right. Now, if you're alert at this point, you'd be saying, but one of the differences between development and ecological dynamics is that development always sort of produces the same form, whereas ecological dynamics seems to generate a lot of variability. So when NASA started taking animals into space in the 90s, they took vertebrates and invertebrates and breeding them to perform a variety of experiments, one thing that is quite striking is that they look exactly like animals in the terrestrial environment. So changing the strength of the gravitational force doesn't fundamentally seem to change animal morphology or function. So, whereas if it was totally selective, you might think, well, surely there would be some changes if it was a purely selective rather than a programmed development. Is this clear? Maybe gravity is not sufficient. Maybe you have to change many parameters. So what you have to explain then is why development looks algorithmic. Why is there a discipline of development? Why wasn't it always called ecology? And it's because there's something about development that looks different. Right? And this is actually some work that I did with Miguel Fuentes, who some of you perhaps know, will talk next week, and a student of ours at Stanford who works in a stem cell lab, who's now at medical school. A little bit more math, not much. So think again about development like this. You have a cell. It can diversify. This is called differentiation in development, or it can replicate. That's called proliferation. So it has a decision at any point. Make a different kind of cell, or make a copy of the same cell. Okay? And you do so at a certain rate, given by these rate constants. Now those rate constants are parameterized. For those who are not really interested in the maths, here's the key. You can either have an internal program which tells you what to do at every cell division. So after the first division, I proliferate. Second division, proliferate. Third division, differentiate. Or I can say, wait a minute, what does the other cell say I should do? I can listen to my environment. So there are other cells dividing and proliferating, and they're telling me what their state is through signals. So Pablo can say, I'm in state 4, I think you should be in state 4. So that we call somatic selection. So that there's a selection pressure, meaning something that biases my proliferation and differentiation. So I have two strategies: ignore everybody, or listen to everybody, and come up with a kind of wisdom of crowd solution to what the body should be doing. And the question we were interested in is, development looks algorithmic. It has this feature that the cells seem to do what they want to do, somewhat independently of what the other cells are doing. Okay? Because you don't want a cell in your heart to listen to the cell in your kidney, which says be a kidney cell. That might be an error. Whereas in ecological systems, you don't have that degree of internal predestination, right? Because you have a selection dynamic which gives you the correlations with the environment. You listen to the environment, right? So you do a little bit of maths, and you make the following assumption. You say, imagine development lives on a manifold, on a surface, and it follows a trajectory. These kinds of illustrations were made popular by someone called Waddington. But sometimes you get perturbed from the trajectory that you want to follow, and you follow another trajectory. Let's call this one a bad trajectory, deleterious. So what development wants to do, either by selection or by programming, is make sure you go this way. Now notice that by selection there's a problem, because if the environment changes, you don't know what's right and wrong, right? It'll push you over here. So you define some functions which tell you how much you deviate from the desired trajectory. So that's all these things are. These say, how much does the development of your process, which is measured in these continuous variables, deviate from the error-free case, r equals 1? I mean, again, I'm just throwing these up to show you that there's a little bit of work here. But conceptually, I think it's quite clear that you're trying to minimize the deviation from a preferred path. And there are two strategies, which I told you before. One is programmed, and one is selective. The selective regime listens to the environment, and the programmed regime ignores it. Just endogenously specifies the program. And you define this function which tells you the difference between the two strategies. All will become clear with the next picture, I hope. So, here's an error rate. Development with no error, development with lots of error. And here's a strength of competition between cells in the body. And what we found is the following. If the error rate is low, so cells, when they divide, they don't make mistakes, okay? And the competition between cells is weak, right? That's when you want selection. So the brain, for example, in the formation of the brain, it chooses a much more selective type approach to consolidating connections. However, when the error rates are high and the competition between cells are high, then you want programs. Because what you don't want is someone who's wrong to tell you what to do. Because you have lots of competition and the error rate is high, and I'm listening to Pablo and he makes a mistake, says, no, you really should be a kidney cell. I really don't want to listen to him. So, here's the point, right? High competition and high error favor program development. So your bodies, somewhat paradoxically and anti-Hamilton, are at the most competitive place on Earth, much more than an ecosystem. So there's this idea by kin selection that all of your cells don't compete. It's the opposite. Your cells will not tolerate deviation from a preferred trajectory. They will outcompete any cell that misbehaves. It's the most competitive ecological arena on Earth of an organism. And again, this one, somewhat counterintuitively, lower competition and lower rates of error give you this Darwinian arena. Okay. So just to recap on this, let me do that. Try to tell you a little bit about niche construction, that the environment doesn't come for free, and that it changes the number of species that you can support in equilibrium. But in fact, developmental variability can be viewed as a form of niche construction. But what makes it look different, and why it has a different discipline associated with it, are factors that are actually ecological: competition, error, and noise. And you can show why development looks programmed versus selective. So we have these bodies by virtue of monopolies, and very strict competition between cells, at fairly low rates of error. That's why bodies exist. You get rid of the monopoly, no bodies, no organisms.
So is there a connection between both program and selection mechanism?
Yeah, because it's a continuum. So every cell wants to do something, wants to divide into a successive state, but the rate at which it will divide into that state is going to be dependent on the state of its environment. And that's the standard ecological dynamic. But under certain conditions, it stops listening to its environment, and it puts much more of the information into its own code. And in some limit, it becomes deaf. So if the whole ecosystem were an organism, you could still get diversity and complexity of some kind, but it would all be endogenously driven.
Right, it looks like it might be possible to, if you are programmed, at some point you minimize or you decrease eventually competition and the rate of error, so you might foster the takeover of the selective.
Well, that's absolutely right.
It's like
that's some kind of cyclical.
Right.
No, I think that's absolutely right. In fact, okay, the clearest example of that, by the way, which demonstrates the importance of competition in bodies, is cancer.
Right.
So people say, well, there's no competition between cells. Well, there is, because as soon as cells stop listening to what their other cells are telling them, they take over, and you get a cancerous state. So a cancerous state is actually a shift between the programmed and the selective regimes. So the cell locally is doing what it wants to do, but with respect to the aggregate, is now in the wrong stage. And so I think cancer is a very good example that demonstrates that the standard perspective, that the body is not an ecosystem without competition, is wrong. Because all you have to do is lose the ability to listen to other cells, and you take over. So there are instances which are on the path we're describing, but not fully there.
So could you say that organisms that don't get cancer are more immediate in terms of...
Yeah, they're subject to the tyrannical dictates of the monarchy, of the germline. Yeah.
Yeah, but I was thinking in terms of some kind of revolutionary control over this.
Well, all organisms get cancer, right?
Plants don't.
Well, that's debatable.
Oh, really? Okay.
Yeah, and so it's what I mean. One of the problems is metastasis. Is that
Yeah, crazy growth.
right? So you could have local proliferation of tissue in plants, like in galls, which are a sort of induced cancer, right? But they're not metastatic. But the basic phenomenon of the loss of developmental control is probably
Okay. Right.
all right. So again, is this all sort of pretty obvious what's going on here? That you can't, I think for me, you can't take for granted the diversity of an ecosystem. And as a theorist, you need to sort of explain it from the bottom up. And niche construction gives you a way of explaining where the ecosystem came from, ideally, instead of assuming it was there. But then it gives you this other insight that in fact you can't assume that there's an environment in an organism, because in fact there's a continuity. And that the organism is a special part of the ecosystem which circumscribes its control locally. That's the difference. Otherwise it's just a continuum. Okay. Now, this approach to development is very simple. There's no genetics in it, for example. Purely ecological modelling. So the question now is, can you do better? Can you go down to genetics too? And this would be more complicated, but I'm going to give you some insights here. So I've told you a little bit about this very simple idea about development, is that you have germlines and they use resources to make soma, stoichiometrically. That's what you do. But where's the genetics? Well, most people think development is the interaction between genes, transcription factors, structural genes, to make phenotypes, make proteins and tissues and organs and bodies. So you need that kind of model too. Now, this is Stephen Jay Gould, who's no longer alive, but he was fascinated in this, the implications of the developmental program for evolutionary dynamics. He thought that it had been hugely undervalued, that constraints that come out of the construction process exert a very significant influence on the evolution of life. Okay? So whereas Darwin said, given enough time, the correlation between the organism and the environment will become almost perfect. Gould said, maybe not. Maybe the construction dynamic imposes constraints such that never can be true. And he made a number of statements, I'm going to go through them. Firstly, this has been noted by many people, the so-called sparseness of the morphospace. There are many more forms of life that we can imagine than we observe. Why is that? Why isn't it, over the length of evolutionary time, just filled in continuously? Why isn't there something that is a kind of missing link of morphology? Right? Why is it discrete? The big problem. There are theories that try to address it, we can discuss it during the week, but it's kind of a non-answered question for me. The other one that he was interested in is the so-called violations of phenotypic isotropy. That is, variation doesn't seem to be symmetric. Right? Variation doesn't, imagine a plane where you had 2 dimensions of character. You started here, you might imagine by mutation that you move out radially. So you sample the trait space uniformly. But in fact it's not true, it's more like this, it's more elliptical. There are favoured dimensions of variation. And he was very interested in that, because of course if there are favoured dimensions of variation developmentally, well then selection can only work on variability, so there will be favoured distributions of species. Over time, there are deviations from what's called uniformitarianism. Charles Lyell, who was also mentioned by one yesterday, is famous for this concept. And this is the concept which says that processes of environmental change are time invariant. So it's like a homogeneous Markov process. Everything we see around us today is a function of forces that we see today. Okay? It's just given enough time, you'll converge to a particular form. But you don't have to invoke differences in time. So the explicit example, the Grand Canyon in North America, you'll say, wow, that must have been a pretty catastrophic event that generated it. And I'll say, no, you just have rain and you have wind and you have a lot of time. And Darwin appropriated the concept of uniformitarianism for his theory of evolution by natural selection and said, enough time with small changes will give us everything we need, whereas there were others at the time, the most notable proponent of catastrophism was Cuvier, who said that's not true. So the early Earth was very catastrophic, and that generated lots of variability, and then later it calmed down. And Gould is a catastrophist. Gould said, I don't think it's true. I think evolution, the clock, doesn't run at a constant pace. Sometimes it runs fast, sometimes it runs slow, and so, in consequence, evolution changes its character, what's called its tempo by Simpson. The observation that convergence is much more frequent than we assume. Does everyone know that convergence is like different lineages having the same structures, like wings, or the vermiform structure of snakes and worms? It didn't inherit that structure, the conversion. And this is a slightly more recent and slightly more technical remark, and that is that all of this stuff is guided by what are called regulatory genes rather than structural ones. And I'll talk more about this in a second. I mean, most of you are biologists here, so this means something to you when I say regulatory versus structural gene, or not? No, I mean, I don't know. Just tell me, I'm not offended, don't be shy. I mean, it's fine, right? If you don't know, it's fine. So, just to sort of calm things down a little bit, the clearest example of the kind of Gouldian worldview in practice is in the TV series Star Trek, where whichever planet Captain Kirk goes to, he can always find someone to have sex with. It's actually a very interesting characteristic of Captain Kirk, right? And because they're all basically humanoid, right? They're all bipedal, they're all vertebrates. They all have 2 eyes and 2 legs and 2 arms, with a few exceptions. Now, I've shown this slide before at talks, and I'll tell you that I will buy 2 beers for anyone who can name all of these species, and I'll buy 1 beer for anyone who can name these. When I give this talk in Santa Fe, one of the girls in the audience could name 6. Yeah. It shows you how we spend our time, right? But I'm sure how many, can you name this crew? I'm very sort of pessimistic about the Chilean contribution to the Star Trek universe. And so, but can anyone name even a single species on this slide?
But it's not that easy to see them.
What do you mean? Maybe there's too much light.
Let's start with
No, this is a first. You guys can't even name one. This is just horrifying. So,
a Krakauer.
yeah, yeah. Well, I'm going to tell you, there are very few exceptions to the rule in Star Trek that morphologies, wherever you find them in the universe, are fundamentally anthropoid. And it's kind of interesting, right? And it's something to do with empathy, presumably. But there are this one, the Davinian neural parasite, the tribble. I mean, most people know tribbles, for Christ's sake, and the horta. I want to tell you guys that at least 50% of the audience in Santa Fe knows those. You need to go and rewatch Star Trek. And there are 500 species in the Star Trek universe, and I highlighted the ones that you just saw. And many of them have this anthropoid property. You will look horrified. You're probably all reading Proust rather than watching Star Trek, which is a little bit intimidating. All right. So, let's go back to something more serious. The question is, what makes the Gould doctrine, or the common structure of the Star Trek universe, likely? So I'm now going to tell you that maybe it's not as stupid as it seems. Okay? So this is how you might write down, a little bit more formally, a regulatory network in genetics. And there are many ways to do it. those of you who come from mathematics and physics and engineering will recognize this as essentially a perceptron or a single layer neural network. It's just linear algebra that I'm showing you now. So what you have is you have transcription factors. These are the things, genes that turn other genes on and off. And they bind to the promoters of structural genes. So here you have a number of transcription factors, and here you have a number of structural genes, and they bind to them with different affinities. And if they bind very strongly, the gene gets turned on. If they bind weakly, it doesn't. And so you can write down a very simple, you can generate a random matrix for development. You'll generate something like an Erdős-Rényi graph. You just assume there's a constant probability of it being occupied. And then you just multiply a developmental matrix, right, by the transcription factor vector. You push it through a nonlinear function, and you derive a phenotype vector. Okay. So matrix multiplication with a nonlinearity. So income, transcription factors, development, outcome, phenotype, and they're both represented in this binary vector space for convenience. So you can imagine what I just told you, what would be called a single projection, a mapping from genotype to phenotype, which is the mapping from one vector to another. Okay? Or you could actually feed the output back into the input. This is called a recurrent design. Okay? This requires that the developmental matrix is square, the dimensions of the vectors are the same. Or a recurrent random projection where the developmental matrix is no longer time invariant. It's a non-homogeneous Markov process. We don't care about such things. So you can write down models, and these have been extensively explored now by systems biologists, geneticists. One of my colleagues, Andreas Wagner, has worked a lot on these models. It's perhaps the most popular model for exploring developmental dynamics. What are the consequences of writing down these models? Now, a number of slides. The basic point is interesting and kind of obvious. I'm going to define a regulatory dimension. The regulatory dimension, right, is just, if you like, the number of bits in the transcription factor vector. So I told you this transcription factor vector, it has ones and zeros and so forth in it. Okay? And the number of elements there, let's call that R. So that's the regulatory dimension. And as you increase the regulatory dimension, the number of possible phenotypes, when you multiply that by a matrix, imagine you multiply it by the diagonal matrix, is going to be 2 raised to the power r, because it's a binary vector. The total number of vectors is 2 to the r. And so this is the number of phenotypes, vectors that you could generate as you increase the number of bits in the transcription factor vector. But if you actually look at the number of vectors that you generate, right, it's much, much less. Okay? And so this is the regulatory dimension, divided by the total possible. The observed divided by the possible, 2 to the r. And what you see is, as you increase the regulatory dimension, the percentage of phenotypes that are visible, expressed, drops systematically. Okay?
So when you say visible or expressed, are you talking that you have data that you somehow...
So what you do is you look at this model and you calculate, you enumerate over all, let's say, 2 to the r input bit strings. You throw them through that simple mapping, and you get out phenotypes which are binary vectors, and you count them.
Okay.
You know if the developmental matrix was diagonal, and right, the square matrix, that they would be equal. You have the same number of phenotypes as genetic configurations. But it's not true, right, for non-diagonal matrices. And it turns out that for non-diagonal matrices, you have very few phenotypes.
Okay.
And that you get fewer and fewer and fewer the higher the dimension. You get the one. That's the first critical point. In fact, and this is the only power law in the talk, you can define a neutrality level. And what neutrality is, it shows you basically, this is the mapping, it's These are one-to-one mappings. The number of a unique genotype generates a unique phenotype. Over here, right, you have many, many genotypes that make the same phenotype. Okay? So on the left-hand side, one genotype, genotype 1, maps to phenotype 1. Whereas on the right-hand side, these are all these cases where you have multiple genotypes, and they all map onto the same phenotype. So you'll notice over here that you have cases where you have over 1,000 genotypes all mapping to the same phenotype. That's the consequence of a developmental map. It compresses the image of the pre-image. Right? It goes from a volume in this n-dimensional sphere, in your vector space, it's mapping between two spaces. It goes from a big one to a little one. And this is just illustrating the point further. And what's interesting about it, look on the right here, this is a phenotypic network. Each node here is a vector in phenotype space, and the radius, right, is proportional to the log of the frequency. So the more phenotypes, the bigger the circle. And what you notice in this space, it's not a genetic space, it's a phenotypic space, is that most of the most common phenotypes are neighbors. So a very interesting property of these developmental maps is that most of the phenotypes that you see in the world should look like each other.
What does it mean to be a neighbor there?
Because you can define phenotypes in the binary vector space, it's Hamming distance. And so two nodes are connected when they differ by 1 bit. Jeff?
Do the phenotypes look like because of developmental reasons, or is they're exposed to similar environments?
There's no selection here. Purely because of the properties of pleiotropy and epistasis in the developmental matrix. There's no selection. Yeah, there's nothing.
Those are just visible phenotypes?
Mm.
Those are just
Visible.
visible phenotypes?
Visible.
Yeah.
Yeah.
So
Because there are many.
yeah,
Of course, because most of them are visible. Yeah, these are visible phenotypes. So, again, I was going to emphasize, we're trying to answer some of those remarks that Stephen Jay Gould made about biases in the developmental plans that are selection-free, that come from constraints. And this is just an illustration of the principle that if you do any projection through any dynamical system, and this is a very simple one, and I'll show you it gets much worse, you have to get what Stephen Jay Gould was suggesting. And for those of you who don't know about evolution, the common assumption in evolutionary dynamics is that this is not true. That all genotypes can be accessed. The only constraint is the probability of move. You're more likely to move to something more similar to you. But there's no fundamental constraint in terms of the occupancy of morphospace. But as soon as you put a developmental plan in, it has to be true. And it's true in a way that's very similar to what Gould intuited. That's just, yeah.
Sorry, one more question. So do the dynamics of modeling constraints are fundamentally different from the dynamics of modeling selection in the conventional sense?
Absolutely, absolutely.
They're fundamentally different.
They're fundamentally different, yeah. Yeah. Is this clear to people? I mean, just to give you a certain intuition, is that when genes are interacting, when I interact with Pablo and Pablo interacts with Jessica and so on, that starts imposing constraints on our degrees of freedom. So before we could move anywhere we wanted, now we can't. So any model that you create is going to have this restriction effect. What's interesting about it is it's restricted in a way that's structured. The common things are like each other. That didn't have to be true if you hadn't written down the mathematical model.
So, I was wondering if you can have the same effect, but from a different principle, just because of you increase your number of transcription factors,
Yeah.
and if you put some noise,
Yes.
noise will mean that error will start creeping, so that will eventually limit, like an error catastrophe, kind of, or error threshold
Yeah.
kind of idea.
I'll talk about that a little bit later today. Noise will tend to weaken this signal. Now, these are random matrices, these are average statements. But anything that makes the mapping less exact, anything that perturbs the D matrix will tend to eliminate the effect. So there is, all of these have to be, I guess what you're saying is all of these have to be compared to a null model. But this is already a kind of null model with respect to selection, because there's no selection. But I'm not sure, since these are statistical averages, I'm not sure noise would really do
Because the
much.
larger the vector of transcription factor regulating development,
Yeah.
the more likely that you will commit more errors, so that will limit the number of
Yes, but-
possible phenotypes that are being
Well,
realized.
right.
Maybe.
That might be
Yeah.
true, but again, the more the higher the dimension, the higher the density of your developmental matrix, and the stronger the effect. I think there's no way of getting away from that. So this, by the way, predicts things like this. It predicts that organisms that have a higher regulatory dimension, like us, should produce offspring more like us than bacteria. Because one of the mysteries of heredity is why we produce offspring that look so much like us. Why doesn't my offspring look like another species? We have most of the genes with others, we share most of our genes with other species. And the argument here is that actually it's because of this kind of bureaucracy of the developmental matrix that the constraint is so severe that you can't escape from that local space. Now, in some audiences, they'll say, a single projection is too simple, those more interested in mathematics. You have to look at recurrent projections. You have to look at the dynamical system, not just linear algebras of motion. And I'm not sure how technical this audience is. I'm going to go through some things very fast for the one or two of you who are. Okay, so apologies to those who are not. It's been known for a long time that when you write down models like the one I just told you about, there are a certain class of logical functions that it cannot compute. So if you have a single projection mapping, it can solve logical problems in the class, in a simple class of Boolean functions like the OR and the AND function. Can't compute the exclusive OR. How many of you actually have any knowledge of these kinds of things? None? Oops. Okay, it only gets worse. So this is something you absolutely should know, by the way, irrespective of what I'm telling you. And that is that it's quite interesting. Does everyone know about OR and AND functions? So, surely. You know about these. So basically, there are truth tables that have all worked out, right? Here, let's get up. And when you write down your inputs would be like 1, 1, 1, 0, 0, 1, 0, 0, and you write down a truth table. Right? So the OR function would give you a 1, 1, 1, and a 0. Because you can have a 1 or a 1 gives you a 1 or a 0 gives you a 1, 0, 1 gives you a 1. Right? The AND function would give you only a 1 here and 0 everywhere else. Okay, because you have to have both bits on. Right? And the exclusive OR has this property. And so this is the basis of all digital computation. And so now, if you think about it on the plane, 0, 0, 0, 1, 1, 0, 1, 1. With a single line, I can define both the AND and the OR function. Here. But how can I draw a single line and generate the exclusive OR? You can't. There's no single line, or in higher dimensions, hyperplane, that can separate the points. And so for this, you need two, this is called polyhedral separability. And so if development has transcription factors as input, so this is a vector with two elements, 1, 0, 0, 1, so on. Higher dimension, and it needs to create different states of the organism, right, in response to those inputs. Okay? And they have this feature that they don't conform to very simple basis functions. Then development has to do polyhedral separability. And polyhedral separability, for those who are interested in such thing, is NP-hard. So you can show the so-called K separability of these K hyperplanes. Does everyone know what? Do you know what NP-hard? Do you guys know about computational complexity? So NP-hard means that it's basically a random search to find a solution, but validating the solution takes polynomial time. It's very quick. So once I have a solution, you know it's right or wrong. You tell me, oh, that's right. But finding the solution takes forever. So if development has to do polyhedral separability with these recurrent networks, it's actually a random search. So another tool would be to show you that development uses so-called approximate algorithms, which don't have the NP hard problem. I mean, that's another tool. Final remark for the even more sophisticated of you in this domain is the so-called Cybenko theorem. And this is that sums of sigmoids, this is the definition, are so-called dense in the unit square. This means that this is a universal function approximator. This means that a two-layer developmental plan, or perceptron, is a universal computer. It can calculate any function. So any categorization of the kind that I showed you up there can be performed by a developmental plan with two projections, any. And this is proven. So it's kind of interesting, by the way, that in development you do tend to have at least two, right? You have transcription and translation. So an interesting sort of philosophical aside is whether or not that was critical. Because with a single one, there are quite elementary categorizations of the inputs that you could not separate. Okay, and the final one for the statistical physicists, I'm trying to appeal to every one person, is the so-called random matrix theory, which you might be familiar with if you do statistical physics. There are some very interesting and hard problems in random matrix theory that haven't been solved that we need to solve some of the problems that we're dealing with now. Because most of random matrix theory comes from sums of random matrices, and we're dealing with products of random matrices. And because matrices multiplication does not commute, it's very hard to do analysis. And so there are some really hard technical problems in doing mathematical developmental biology because we don't have the tools from statistical physics that we need. So there needs to be development in random matrix theory. For the one person or half person in the room who's interested, we can talk about it afterwards. The consequence, though, of all of that stuff is it makes it worse. And so the proportion of visible phenotypes when you have recurrent plans actually goes down way below the single projection. And it's worse for the random non-stationary markup process than stationary. It doesn't really matter. Again, the message that I told you remains true, it just gets worse. Too much. All right. Final point. Let's imagine an evolutionary sequence. You have a bunch of genes, and by duplication, you take some vector in the developmental matrix and you duplicate it. So a standard assumption, by the way, in evolution is that evolution proceeds by duplication and deletion. So you take a gene, you copy it, it's neutral, maybe it does something new, you keep it, or if it does nothing, it just goes away. It's a pseudogene. So you could take our model and you could actually subject it to an evolutionary dynamic. Right? Where you duplicate genes, make the dimensions of the D matrix bigger. And if you do that, you have the following observation. And I'll just stick with the observation. Imagine make a phylogenetic tree of all phenotypes in the world that come out of this dynamic. And there's an ancestral developmental matrix, and it produces descendants, and they produce descendants. So the black square on this tree, which is a phenotree, it's a tree of phenotypes, you'll notice are dotted all over the tree. Whereas the descendant species are localized to branches. So the ancestor spans almost the entire morphological space, whereas the descendants are more concentrated. So I told you earlier that this follows from the increase in the regulatory dimension. Low dimensions, you get a huge amount of what gets called by evolution biologists as disparity. That's the embedding of diversity in the metric space, distance between things. But later in time, you don't get as much disparity. You get a more diversity because you produce more species, but the species are all the same. So this is the first and second principal components through the phenotypic vector space, where the axes of the ellipses are the mean and the variance. People here know about factor analysis. Okay. So the point being here again that in the volume, in the principal dimensions of phenotypic space, the volume of the ancestor is larger than the volume of the descendant. And I mentioned this again back to Gould, if you read his book called Wonderful Life, one of the points that's been made extensively about the Cambrian radiation is that you get a lot of phyletic diversity. So a lot of species came into existence there, forms came into existence. And later in life what happened is we just filled in locally the variability, but it spanned a huge amount of the morphospace. Okay? And this is completely consistent with the idea that early in life you were living in a time of lower regulated dimension. And that as you tuned regulation to make yourself more responsive to different environments, you also imposed more constraints. And so early you have phyletic diversity high, but species diversity low, disparity high. Late you have species diversity very high, but phyletic diversity relatively low. Now, nearly finished. Jess and I went to visit a friend of ours, Doug, who I mentioned at the beginning, Doug Owen, who is the curator of the Burgess Shell at the Smithsonian Museum of Natural History in Washington. And we were very lucky because we could go and look at these things, and it's not easy to see. And you expect, when you go and see the Cambrian fauna, right, to see beautiful, amazing fossils. And of course, this is what you see. And it's like, Doug, where is it? Where is that fossil? Well, you see, this is really that. And if you just sort of rotate it a little bit in your imagination, so, you know, biologists, like archaeologists, can turn a single rock into a pyramid, right? It's like, but anyway, it's a bit disappointing for us, because we expected something more grand. Let me just make one final point about this being a general principle. This looks a lot like a statement about development. For those who don't care about biology, those kinds of models I showed you could be quite general models of regulation. After all, neural networks were invented to study brains, not to say development. And I'm going to show you this because I think it does actually apply to cooperation. And this event relates to a discussion we were having at dinner. So, who are these people? Come on. Who's that? Who's that? Huh? Oh, fail. Okay. Paul Allen. Paul Allen. Who's that? Yeah, and okay, good. Thank you. 3 out of 4, not bad. Now, around the same time, you build companies, and what's the difference between these companies? Well, one difference back then was the size of it was effectively the revenue dimension of Microsoft versus Apple. Microsoft is a very large firm with many bureaucrats. Now, by our theory, what does that mean? It means you can produce many types, but they're all the same. That's what it says. Right? When you're smaller, like this company used to be, you can produce fewer types, but the types you produce are very different. And we all know that's true. If you work in a large company, of course you can make more product, because everyone has to produce. But everyone is producing exactly the same product. And it's because of the interactions. You're not allowed to innovate. So I think that the principle is very, just like I mentioned, is very generic. And we see it wherever we look. And it's one of the reasons, by the way, why the Scientific Institute consciously decided to remain small. Because we have to keep a small bureaucracy so as not to converge in this smaller volume of solutions. And so it is a general ergonomic principle. Yes, yeah.
So you're making a point about the distinction between diversity and disparity?
Yeah, but diversity and disparity, because thanks. So again, diversity just counts the number of points. So the number of points will always increase as you increase the dimension. But they're all getting more closer in an embedding. So yeah, one is a measure of just a number, the cardinality of that set, and another is actually a statement about metrics, distances. Complexity, by the way, which will be later, so diversity number, disparity, typically a continuous measure of similarity. So now it could be KL divergence, entropies, and so on. Complexity is usually a measure either of the time it takes to make diversity, right? The compressibility of your diversity, factorizability of it, or the description of the program required to generate your diversity. So there are 3 classes of complexity measure: time, informational, right, in terms of compression, and algorithmic. Okay. And so I haven't talked about it, I'll talk about it a bit later. I think that's that. Summary, yeah, quickly. The construction dynamics, a very general approach to diversity. Okay. Shows you how you can get more of it ecologically, but it also it sort of integrates a little bit across development and ecology. But I showed you that there was a fundamental dilemma that it's not, I didn't go into game theory, but you can show that it's not an ESS, it's not a Nash equilibrium in the game. It can be algorithmic or Darwinian on a continuum according to the strength of competition and noise. So development looks different from ecology because it's fundamentally different for these reasons, in part. That once you have construction, it has imposes significant constraints on variability, along the lines I just told you. And that the more complex the construction, the more evident its footprint. So at a certain point, right, when a bureaucracy becomes so big, all of the structure is the bureaucracy rather than the constituent contributions. So that's Microsoft. Now it would be inappropriate of me to talk about complex systems without talking about von Neumann, because he's one of the key figures in the history of the field. And in the 1940s, von Neumann is very important, certainly in mathematics, mathematical physics, quantum mechanics, co-inventor of the digital computer, worked on the Manhattan Project. Von Neumann was became interested in biology, and in particular in replication. And very quickly he realized that replication is a trivial problem. It's very easy to make something replicate. And again, we'll talk about this later. So he became interested in what he called his universal theory of construction. So in 1948, he gave a lecture at the Institute for Advanced Study on the theory of universal construction, where he suggested that all living systems have to have three components. And just to show you what von Neumann was saying, and he wrote down an algebra of life. Okay? And the algebra of life, he said there are three components. There's a message, there's a structure, and what you need to do in living systems is that messages need to be replicated, two of those, and you need an automaton that does it, and that messages need to build structures. Okay. But you need to decide which of those automaton to activate, and that requires a third automaton that moves between replication and construction, proliferation and differentiation. It's a proto-niche construction model. He didn't articulate it in those terms. And so if you think, all living systems are then constituted of a structure plus a message. And the key for von Neumann was to cheat, or not to cheat, he said equal to the vector A, B, C. And this is a self-replicating constructor, because the vector it includes in it are all the components necessary to replicate and construct. He wrote down a formal mathematical model for this. And later in life, as Freeman Dyson got very excited about, when he wrote about this kind of stuff, he realized that in some sense, yes, so this thing we'd call something like a polymerase. Now this would be the ribosome, right? This is a ribosome. This is a polymerase. And this is a transcription factor. So in some sense, a little bit like Schrödinger in Modern's life, von Neumann had algebraically intuited the tripartite constituents of all living systems. Stan Ulam, who was also working at Los Alamos at the time, showed von Neumann how you could turn this into a cellular automata model, which I imagine Eric has talked to you about. And he wrote down the specification of the first self-replicating universal constructor cellular automaton. So I think it's very that this theory of construction has been there all the time since the 40s in the background as the critical theory that we need to develop to solve problems of living systems. And it's interesting how it's been sort of neglected. This died, it sort of didn't really go anywhere. And it's not clear to me why, actually. Von Neumann moved on, worked in quantum mechanics and so on. But it would be inappropriate not to mention it, and so this is just a schematic that he drew for his constructor. So here's some references. You can download all of these from a web page if you want to. And later I'll talk about complexity. Thanks.
Thank you.